Good morning. In today's lecture, we are going to study aseptic techniques. So, what is aseptic techniques? So, that aseptic techniques are the techniques or procedure that is done to prevent the contamination from pathogen. So, what are the pathogen? That are unwanted growth of microorganism. So, that can be fungi, that can be viruses or bacteria. An aseptic technique also termed as sterile techniques. So that's mean to maintain the sterile condition to prevent the contamination. So why those are necessary? So as we are studying the plant tissue culture, for growing this plant we are going to use medium that contains sucrose, micro and micro elements, vitamins, some growth hormone. That is the source for the growth of plant as well as microorganism. But we are interested in plant, so we want to prevent the growth of microorganism. But what is the problem? That can grow, the microorganism grows faster than plants. So that's why there is importance of aseptic condition during the culturing. So what are the sources of contamination? The culture vessel, right? That can be glassware, plasticware, different instrument like scalpel, blade, then nutrient medium, that is media, then instrument used during various operation, then explant, that is plant material, and then operator, the person who is performing the procedure like media preparation or inoculation of plant into the medium, then environment of the tissue culture laboratory. So these are the sources of contamination. So we will start with the aseptic techniques from the glassware and plasticware. So to maintain the aseptic condition, it is very important from the washing step. So to wash the glassware and plasticware, the different detergents are used. So you are going to tell me why the detergents are used for glassware and plasticware. What is the reason behind it? So how you are going to do that? So first you are going to wash that glassware and plasticware thoroughly with tap water, and then make the detergent solution and soak this glassware and plasticware into that detergent solution for overnight. And then after overnight soaking, you are going to rinse that glassware and plasticware with tap water, and keep in mind that tap water should be full open. And after tap water washing, you are going to wash the glassware and plasticware with distilled water, right? So after washing, what you are going to do? You are going to So after washing, you are going to place that glassware and plasticware into the trays or basket to allow the maximum water drainage, right? And after that, you are going to dry the glassware and plasticware in the oven or hot air cabinet. So this is the second question. What is the principle behind drying the glassware and plasticware in the oven or hot air? And after that, after drying the glassware and plasticware, you are going to store that into the dust proof cupboard, right? So sometime what happens, the glassware has dried agar, right? So you cannot use that as it is, right? So you have to clean that agar. How you can clean that agar? By autoclaving. So what happens by autoclaving? That temperature, the agar will melt. And when this glassware used for the culturing or any other purpose, so it is done. So you are going to wrap that glassware with the cotton if it is a flask, right? So after wrapping with the cotton, you are going to wrap that with aluminum foil and then the paper. Keep in mind, right? So in like microbiology or any other experiment, what we are going to do? So first we are going to place the cotton in the mouth of any glassware. Then we are going to wrap that glassware with the only paper. But in case of plant tissue culture, we are going to wrap with first foil and then paper. or it is good before the wrapping with the aluminum foil. We are going to wrap with the paper and then foil, right? So next is, other than glassware and plasticware, we are going to use the forceps, scalpel, needles, spatula, right? So that is used during the trimming of explant and inoculation of explant into the medium. So how that can be sterilized, right? So that can be sterilized. You are going to wrap the forceps, scalpel, needles and spatula with aluminum foil. Obviously, we are going to wrap first with paper, then aluminum foil, and then autoclave it. And during aseptic manipulation, you have to dip that instrument into the 95% ethanol and flame it and cool. And after cooling, you are going to use for inoculation or any other purpose. Right? So next is, so this is for glassware and plasticware and other instrument washing. And so now we are going to see the media sterilization. So why there is need of media sterilization? As I told you, the media is rich in sucrose and other organic and inorganic nutrients that supports the growth of microorganism, and the growth of microorganism is faster, and that's why it can kill the culture vessel, and that will leads to the failure of culture. So the contaminants may also give out metabolic waste that are toxic to plant tissues. So that's why it is absolutely essential or important to maintain the complete aseptic environment inside the culture vessels. So what are the possible sources for contamination of the medium? So the culture vessel itself, so that's why it should be washed, dried and autoclave, then medium itself, then explant, then environment of the transfer area, then instrument used in handling plant material during inoculation and subculturing, and the environment of the culture room and the operator itself. So that means the students, right? So during practical, you people most of the time talking, right? So that is the source of contamination. So measures to take in the guard the culture. So how you can sterilize the media, right? You first the glass vessel or the medium vessel should be should be closed with the bacterial proof closure, that is cotton. Then it is wrapped with paper and aluminum foil, and after that it should be autoclaved at 121 degrees Celsius for 15 to 14 minutes, right? So this is the method for media sterilization. So you are going to tell me the principle of autoclave. So it comes under the wet sterilization, right? Next is plant material. So that is the source of contamination. So plant material that is used for the explant, right? As the plant grows in the environment, it is exposed to the different condition, right? So you can see it is on the surface of the plant there will be different types of microorganism. There must be the dust, right? So that's why this plant material should be washed, right? And and you can collect the explant from the plant which are grown inside the greenhouse, because if you are going for this method, what happens, there is chances of contamination is less, right? So the source of infection can be avoided through the surface sterilization. So you are going to sterile the plant material through the surface sterilization, right? And so for this surface sterilization, you are going to use surface sterilant, right? You are going to tell me what is meant by surface sterilization and surface sterilant, right? Those are the different chemicals which can be toxic to the plant, but it is important to sterilize the plant material, right? So so that we can avoid the contamination. So what for that, to avoid the toxicity to the plant tissues, the concentration of the sterilant and the duration of treatment is very important. So that can minimize the tissue damage. So these are the different sterilant. For example, you are going to use 0.3 to 0.6% of sodium hypochlorite treatment for 15 to 13 minutes. That will decontaminate the most of the tissues. But you have to keep in mind that is the concentration of sterilant and the treatment of the duration or the time of treatment. So how the plant material is sterilized? So first you are going to cut your explant from the mother plant, then take all the explant into the beaker, then wash it with the tap water, right? Then add the surface sterilant you are wanted to use. Then by washing with the surface sterilant, then decant that surface sterilant and then add the distilled water, right? Then wash it with the distilled water. Then again transfer this explant into the laminar airflow. Again in the laminar airflow you are going to use different surface sterilant, right? For example, mercury chloride, that is the treatment is done for 3 minutes. After washing with or after treating with mercury chloride, you are going to decant that, then give the 3 washes of double distilled water to that plant, to that explant. So what happens? By washing 3 times with distilled water, the traces of that surface sterilant will be removed. Then there are different work station that can be used for plant tissue culture. So that is the laminar airflow, right? Or laminar airflow cabinet. So the area which is present in the laminar airflow, that can be cleaned with the 70% ethanol, right? And after cleaning, you are going to on the switch, and now you know there is UV is the source for decontamination in the laminar airflow. That can be kept on for 15 minutes, and after 15 minutes you can off that UV source, then start the airflow and tube light, and again you can wash, you can clean the surface area with the 70% ethanol. So we are going to study this process in the practical. So you will come to know better, or you will understand better during the practical. So thank you. If you have any query, please ask.